In the current study, we investigated what happens to synaptic connections in inflammatory demyelinating disease relevant to multiple sclerosis using the retinogeniculate circuit. And what we first found is that there is profound synapse loss in this retinogeniculate circuit in early stages of demyelinating disease prior to any significant changes in the myelin sheath or axons. But instead, we found this concomitant with the deposition of complement factor C3 at synapses and engulfment of synaptic material by reactive microglial cells. So we next want to ask in this figure, can we actually protect synapses? So we use a gene therapy strategy to overexpress a C3 inhibitor called Crry in the visual system in this retinogeniculate circuit. And then we found Crry protein was specifically localized to these synapses. And we induced inflammatory demyelination using experimental autoimmune encephalomyelitis in mice, or EAE. And we looked at synaptic changes at the onset of EAE when we see this early synapse loss, and also in peak EAE in a supplementary. And what we find is that there is a significant attenuation of engulfment of synaptic material by reactive microglia with the Crry treatment. And we also saw significant protection of synapses with the AAV Crry treatment. We finally went on to look at visual function using a visual acuity task. And as you would expect with EAE in either animals that were not treated with an AAV or treated with a control AAV, there was a significant decrease in visual function or decrease in visual acuity compared to pre-EAE. And this Crry treatment actually completely attenuated this visual acuity deficit and preserved visual function. So now we've had data that provides evidence of significant synapse loss at early stages of demyelinating disease, accompanied by C3 deposition at synapses and engulfment of synaptic material by microglial cells. And AAV Crry treatment results in Crry localization to synapses, blockade of C3, and blockade of microglial engulfment of synapses, preserving synapse structure and visual function.